This is the online recording for the second XNAT workshop, where we presented integrated pipelines for semi-automated MRI image processing on the University XNAT platform here at the Imaging Centre of Excellence. XNAT is the software we use at ICE for the purpose of storing and transferring imaging and clinical data between the university and the NHS. Our XNAT infrastructure is summarised in this schematic diagram. On the right, in the green box, we have the university instance of XNAT. It is accessible via web browser if you are connected to the university network. Isolated from the outside world is the NHS instances of XNAT here on the left, one for diagnostics and one for CRIF, sequence development studies. The unique selling point of XNAT is that if we want to create a project focused on a specific disease, we could pull data from across the entire network relevant to that disease, making collaborations much more easier to facilitate. What has been missing, however, is a means to process and analyse these MR images within this infrastructure. So what we have done is we've built a new instance of XNAT called Compute-XNAT that lives on the university side of this infrastructure, and its purpose is solely to facilitate image pre-processing and analysis. This works by running another software called Docker on Compute-XNAT. Docker allows us to build bespoke computing environments for doing very specific jobs, and we can deploy these Docker containers from the XNAT user interface each time we want to work with an MRI image. As of the date of the XNAT workshop, we have a number of different Docker containers that we have organized into different groups based on the project or image type that we are working with. So we have a general purpose group that applies to all project types. This includes Dicom to Nifti conversion, co-registration, functions for isovoxeling a Nifti image or modifying a region of interest image in some way. We have a group of functions for working with T1 weighted images, for example, doing brain extraction or creating cortical layer regions of interest. We have functions for diffusion studies, task fMRI, resting state fMRI, and quantitative susceptibility mapping. These functions are all accessible from the main project page within XNAT under the project settings button of the actions page. They can be toggled on and off on a project by project basis, or even a task by task basis if one day you want to work on T1 image processing, or another day you want to work on task fMRI data. The importance of supporting MRI image analysis with semi-automated pipelines can be nicely demonstrated by this 2020 study by Botvinnik, Nazer and colleagues. In this study, they had 70 different fMRI labs sign up to analyse a single dataset. These labs tested 9 different hypotheses, returning their statistics back to authors for analysis. The critical point here is that the different labs all used different methods for deriving their statistics. Different software, bespoke code, different statistical methods. What the authors found was that across the 9 different hypotheses, there were some which had a high amount of variance in the agreement of whether there was statistical evidence to support the hypothesis. In the figure shown here, you can see that there is high agreement in accepting or rejecting the hypothesis for 7, 8, 9, and 5, and that agreement begins to change across labs for hypothesis 2, 3, 4, 5, and 1. And for hypothesis 1, the interval is starting to cross that 0.5 region, so it is almost a bit of a coin flip whether this hypothesis will be accepted or rejected across the labs. This next figure shows the disagreement in more detail for hypothesis 1. This is a Pearson correlation of the statistical maps for 64 of the labs. You can see that 3 different groups start to emerge. The biggest group are all in alignment with one another. A small second group emerges that is anti-correlated with the first but internally congruent with one another, and then a last group emerges that's a bit of a mix between the two but is neither fully congruent with group 1 or group 2. This is the sort of variability that we can try to minimize by using integrated pipelines across the community at ICE, supported by Compute-XNAT and the core support team. The benefits of the XNAT compute infrastructure are broad and will impact different users in different ways. So I won't spend too much time on this slide, but the key takeaway points are that we can reclaim a lot of the time and the cost that we put into onboarding and training new staff members, and we can spend those resources in other critical areas, for example writing papers, writing grants, data quality control, and data interpretation. Complicated MRI image analysis will now be more accessible regardless of a person's experience. I do think it is important to consider the risks of such a system. If it is a resource that many people rely upon for their work and it breaks down, then that will be a huge problem. The beauty of Docker is that the images that make it all work are hosted on cloud storage, and there are many backups. These can be deployed on any machine or high-performance computing system, and they will work the exact same way. We have a backup instance of Compute-XNAT, and the Docker images can even be deployed to other XNAT instances within the overall infrastructure. Compute-XNAT has room to grow. 7T imaging grants supporting XNAT will allow us to grow as demand grows. Another feature of Docker is that the images will always work the same way whether they are deployed today or years from now, here at ICE or on a machine in another part of the world. Over time, the number of functions will grow, either by myself or by users making requests, so development of these tools will grow organically to accommodate the research that is being done in the community. As I have built these Docker containers, I have purposely made every effort to mitigate cases where image processing can fail. There will be edge cases that I have not considered, but again, as these problems arise, that threshold for failure will become smaller and smaller, and the benefit will be for the whole community. All of the functions on Compute-XNAT will be supported with documentation and video tutorials that will be available on our ICE wiki. This is currently in the process of being populated. So we can demonstrate how these Docker containers work by looking at the first example function, the general Dicom to Nifti conversion function. On XNAT, you will have a project, and then within a project, you will have a number of subjects. Each subject will have one or more MRI sessions, and an MRI session will have a number of scans associated with it. The Docker containers are designed to run on scans. So each scan will have a resource. Here the resource is the Dicom folder containing the Dicoms for each scan. The DCM to NEX function expects the Dicom resource to exist. It will target that resource folder and output its outputs to a new Nifti resource folder containing the Nifti images. Dicom to Nifti function can be run from the Run Container drop-down menu. Before it runs, there's a pop-up menu where you can enter command line flags, so the DCM to NEX function. You can run the container by clicking on the blue Run Container button. And when it's finished, it will automatically output the Nifti images to the Nifti resource folder, which it will automatically create. This is exactly more or less how every other function will behave, with the exception being that we might need to do a small amount of setup, as you will see as we go through the different functions. So we will start by looking at T1 weighted image processing. The schematic here is an overview of how we might do this for any given project. So we start with brain extraction, and we give those outputs to FreeSurfer's Recon-All wrapper for extensive segmentation and atlas alignment with our native brain image. And once we have this, we can then do further segmentation on deep brain structures like the hippocampus, the thalamus, and the brainstem using FreeSurfer's automatic tools. We also have functions for creating images for helping us do manual segmentations or helping us correct some of these automatic segmentations. And finally, we have a function for turning any of these segmentations into 3D models for the purpose of data visualization. So the first function we'll look at here is to do brain extraction. We have two flavors of this. Both use FSL's BET to do brain extraction, but we have an extended version for use with the head and neck coil we have here at ICE that gives us a bit more of the brainstem. So to run this function, we will target a scan with a Nifti resource folder, and this Nifti resource folder must contain the Nifti and JSON images for both the uni and the inversion 2 scans. When we run the function, the pop-up window will give us access to variables that BET uses for adjusting the brain extraction function. But the output when complete will be a nice brain extracted image and corresponding mask. So this function works well for diffusion with a bit of an adjustment for the fractional intensity. And if either of these functions have any errors, those errors can be manually corrected in something like ITK-SNAP and uploaded back to the scan Nifti resource folder. And when the function is run a second time, it will use the corrected mask to do brain extraction. The extended version works the same way, except we add additional growing of the mask along the negative Z axis within a narrow image intensity window related to white matter, so that the mask will extend along the brainstem, and we do some filling in of any gaps. And what we get out is a nice brain extracted image encoding the brainstem and corresponding mask. So the next function is T1 weighted Recon-All. It runs FreeSurfer's Recon-All wrapper, and it targets the T1 weighted folder, which is the output resource of brain extraction, and it will place its outputs into this same T1 weighted resource folder. There are no settings in the pop-up window. You can run this function and it will process automatically. The outputs will be placed in the T1 weighted resource folder. Here we output FreeSurfer's subject directory so that we can keep this and use it for future functions. But we also output a number of derivatives that we've selected. So these are organized into a number of selected images, both in FreeSurfer and native brain space. So this includes FreeSurfer's new brain, the ribbon, grey matter, the cortical ribbon, the white matter segmentation, and also the border between the white matter and grey matter segmentations. We also output the FreeSurfer atlases that are implemented in Recon-All in both FreeSurfer and native space. So this includes the APARC plus ASEG atlas, the APARC 2009 plus ASEG atlas, the APARC DKT atlas plus ASEG, the ASEG Auto, NoCC-SEG, ASEG, NW, and PARC atlases. Two of these we also output as individual parcels. So the APARC 2009 plus ASEG atlas. This is a cortical and subcortical parcellation of the brain. It includes 190 different parcels. And then we also output the white matter segmentation atlas as individual parcels. This is 177 different parcels. So once we've run Recon-All and we have that FreeSurfer subject directory, if we want, we can then run any of the further FreeSurfer automatic segmentation functions for the hippocampus, the thalamus, or the brainstem. And again, these outputs will all be placed into that derivatives folder in the T1 weighted resource. And for the hippocampus and the thalamus, we output the parcels as individual parcels again. And this is still to be implemented for the brainstem, as it only contains 4 different parcels. So the next function we'll look at is the 3D model function. For this, we need to create a folder under any scan called 3D model, and into it we want to place a segmentation or an atlas that we want to turn into a 3D model. So here, for this example, I've chosen FreeSurfer's ribbon output, the white matter and the grey matter in native space, along with the automatic hippocampus segmentation in native space. And what you can see in the output, also placed in the 3D model resource folder, is that the function has converted all 3 segmentations into 3D models, placing the output into their own subfolders. This is what the output looks like in Blender. So these outputs are OBJ files, and they can be opened in most 3D modeling software. On the left we have the grey matter, on the right the white matter, and then in red we have the hippocampal subfields. This is another example of cortical areas related to motor processing. The next pair of functions, I call them segmentation helpers. So they help us create images for manually correcting segmentations. They will create for us a smoothed image and a gradient magnitude image of our anatomy. And these are useful for helping us make decisions when it comes to manually correcting things like automatic segmentations. So to start, we create a scoop folder under a scan and place inside it the anatomy that we're working with, either FreeSurfer's or our own native anatomy, and a region of interest that's in that same space. So the first function, create scoop image, will first create a scoop or a sphere around our region of interest. We do this because we want to crop down and focus on the ROI. This is helpful when we're doing manual work. It also helps with file size issues when we move on to the next step, where we want to smooth the image and create the gradient magnitude image at a much higher resolution. But the output for this function is the new scoop sphere image, and it will be placed into the scoop folder. The next function, the GramMag for segmentation, works on this new scoop sphere image. It will crop in on the region of interest. It will upsample it to 0.2mm isotropic resolution. It will run a smoothing algorithm with edge retention, so it smooths the image but tries to keep edges. And then the function will compute the gradient magnitude image on this smoothed image. It will downsample both back to the original brain anatomy image size and voxel grid space, and it will also uncrop the image. So the output for this function consists of a pair of high resolution images at that 0.2mm resolution and a pair of images at the same resolution of the input anatomy. So this image on the right here is showing what the gradient magnitude image looks like in the native resolution. The gradient magnitude image is a tissue rate of change image that can be helpful in determining, for example, the location of CSF in the sulci, or whether a partial volume voxel should be included as grey matter or white matter. But these images work best when they're kept at their highest resolution, where we can easily see where one tissue type is transitioning into another, or whether tissue is transitioning into CSF in the sulci, for example. The next function within this set is the create cortical layers function. Here we are using Laney to create equidistant layers. So the setup for this is that we create a layers resource folder within a scan. We place in this folder the ribbon output from FreeSurfer and a region of interest. And it is important that both images are in the same space. This is what the ribbon output looks like. And the pop-up window will ask us to enter how many layers we want to create. The output for this function will be placed in the layers folder. And what we get here is we get an image that combines all the layers in one. We also get the individual layers output as separate images. And we also get a depth and mid grey matter image. And these are all placed in the layers resource folder. And for this example, creating 3 layers, this is what they look like on the native brain space. So the next set of functions we'll look at will be those that are associated with processing task fMRI. So this schematic gives a bit of an overview of how we might go about that workflow. We'd start by doing top-up to correct field distortions within our image. And then from this, we would look at quality assurance images to make sure that the data we're processing is of sufficient quality to proceed. Next, we'd look at pre-processing our images and then registering them with our anatomy. From here, we can look to do single general linear modeling, multi-run general linear modeling, multivariate pattern analysis, or even retinotopy. So the first function we'll look at is the top-up function. It will run on the Nifti resource, and it will put its outputs into a top-up resource folder. It requires in the Nifti folder the images that we use for top-up. So typically we collect a set of images in the same phase encoding direction as our main run, and a set of images in the reverse phase encoding direction of our main run. And you can see on the example image the effect of field distortions along the phase encoding direction for these 2 images in the anterior-posterior direction and the posterior-anterior direction. And what the function will do is it will find the longest time series image and recognize that as the primary image, the run image. And then it will construct the phase encoding calibration pair of images from the other 2 images. It will build the files it requires from the JSON metadata files. And then we'll run FSL's top-up function to create the image for correcting field distortions. And then it will apply this using apply top-up, again in FSL. And the output we want from this is the data top-up image and corresponding JSON file. And you can see on this example where before and after the effects of top-up make a difference. So you can see where the image is shifted around these red arrows. And in the field distortion map, or the field map, areas where the image is going to move will be associated with really bright areas or really dark areas. So this image can be used as a bit of quality control to see how much your image is being manipulated by distortions and how these are being corrected. So the next function we'll look at is the task fMRI run Laney QA. So this is using Laney to create a set of quality assurance images. This function will run on the top-up folder of a scan, and it will output to a new QA resource folder. So we can run this function after we've done top-up, and on the data top-up image, it will generate a number of different images for quality assurance. This includes the SNR image, the TSNR image. Another pair that are useful are the skew and kurtosis images. Also outputs the mean image of data top-up, which we can use for quicker loading and also to make sure that structurally the image is looking good. These are a number of different examples. From left to right we have mean, skew and kurtosis, and below them SNR and TSNR. And if you'd like to get some more information about how to interpret these images and why they're useful, I would encourage you to go to the Layer fMRI Laney page, where they have a number of different examples of using these images to quality assure your board images. So next we'll move on to pre-processing. We do this using AFNI, and this function again will target the top up resource folder, and it will place its outputs into a pre-processed resource folder. So here the pop-up will give us a number of options for what we want to run. Generally speaking, we would always run slice time scan correction, motion correction, temporal filtering, and then smoothing can be optional, and the size of the smoothing kernel can be chosen here. When we run this container, the output in the preprocessed folder will be extensive. So for each step, we output the corresponding image. We also output the motion correction parameters if those are needed for future steps. Next, we'll move on to image registration. So for this, we again set up a folder, a co-register resource folder within a scan. And the function will target this folder and put its outputs in this folder. So for this, we have two methods. We have a manual method and an automatic method, both using the same underlying functions. We'll start by looking at the manual method. So we have two functions to facilitate this. One is to prepare some images for co-registration, and then once we derive the transform for registration, we have a function for applying the transform. So in the co-register folder, we need to place the data image that we're looking to register and the anatomical image we're looking to register with. And when we run the general prepare ITK co-registration function, we get two outputs. We get a single volume of the data image, and we get an image called new_anat to ref. And what this is it's the anatomy intensity values mapped into the voxel grid space of the data. So it's a low resolution anatomical image in the same space as the data. Now in ITK-SNAP, we can load in this single volume of the data and also add as an additional image the anatomy in the data voxel grid space. And we can see these two images here. You can see that they are quite closely aligned, but they are not perfectly registered yet. So what we want to do is we want to open the registration window from the Tools tab at the top. And here we can define the moving image and the static image. So for this, the static image will be the bold image, and the moving image will be the anatomy image. And we want to do this by rigid body transform, and the metric we're going to use is mutual information. So here we have two parameters we can adjust, the coarse level and the fine level. So this is like a coarse alignment followed by a fine adjustment of the alignment. Because the two images are already quite closely aligned, we can set these to 1 and 1 to start looking at the smallest amount of adjustment we need to make to find alignment. And we can run this registration. And with a bit of trial and error, we can find something that we think looks quite nicely aligned. Now we can use ITK-SNAP's tools to try to help us visualize how well the alignment has succeeded. So here we've taken the paintbrush tool at a single pixel level, and on the bold image, we have marked some anatomical features that have really high contrast. And then we can flip between the two images to see how they look before and after registration. So before registration, you can see that the anatomy isn't quite well aligned with the bold image, but then it starts to look more coherent after registration. And the trick here really is to choose a different number of these high contrast features in the different views. So in the sagittal, the coronal, and the axial view, and with a set of these landmarks, we can then use them to assess how well registration has succeeded. Now, ITK-SNAP also has another feature where we can use a mask or a segmentation and localize registration to just that region. So this can be really useful if you have a region of interest you're interested in. For example, early visual cortex, you can create a mask of this region and just focus registration within this window. But again, if we're using this, we still want to look at local anatomical features to see how well registration has performed. Now when we're happy with the registration, in ITK-SNAP we can save the transform file as transform.mat and upload this into the co-register folder. And now we're ready to run the next function, the ITK apply registration function. And this function will target the co-register scan folder. It will look for that transform map file, and it will apply it to it will apply the inverse transform to our bold data. So it will reslice the data. And what we get out is the data resliced image, and we also output a single volume of the data resliced image. And this can be used with our NAT to ref image to again look at how well the registration has performed. So the automatic version of registration is the function auto co-registration, and it is using the same functionality as the manual process, except it's doing it automatically. So the setup for this function is a little bit different. In the co-register folder, we again have to place the data and the anatomy that we're using, but we need to rename them as target and ref so that the function knows which is the static image and which is the moving image. And for this function, we define the anatomy as the static image and the data is the target image, the one that's being resliced. And we can also place in this folder a region of interest that will act as a segmentation or a mask for that type of image registration. The pop-up window for this function will give you input variables for the coarse and fine adjustments. So we can modify these on a sort of iterative basis if we're working through a number of different attempts. And we can also toggle on and off whether or not we want to output the single volume as a test. So if we're doing this for the first time and we're unsure of how it's going to perform, we can toggle this on to register just a single volume, save us some time, and then once we're happy with the single volume registration, we can then run it for the full bold dataset. The output of this function comes in two pairs, and the first pair are related to the segmentation or mask in the data space, so they can be checked for coherence, and the target and ref is the resliced data, target data in the space of the anatomy. And for these two sets of image pairs, this is what they look like. So at the top we have the anatomy and the segmentation overlaid on top of the anatomy, and then on the bottom row we have the bold image and the same segmentation and mask overlaid on top of that bold image. So the next function, and the first in a number of analysis functions, is the task fMRI GLM function. It expects to run on the GLM resource folder, and will put its outputs in the same folder. So for this we need to set up the folder by placing into it our resliced data, a region of interest where we want to do the GLM, so this can be a small region of interest or it can be the entire brain mask. We also put into reference anatomy so that we can put the statistics in both the data space and the anatomy space. And then we also include a design text file. So this function will auto-detect the anatomy, whether it be brain or new. It will detect data and any other image as the ROI and the design file. It will build an HRF convolved design matrix from the design file, and then it will fit a GLM voxel-wise within inside the ROI, compute contrasts with t-statistics and do multiple comparison correction, and then write the maps in both data space and brain space to the GLM resource folder. So this slide is a bit of overview of all of this in action. On the far left is the design text file. So it's set up where we define a number of conditions at the top, and then we give a name to a condition, a number of trials or repetitions, followed by the onsets and offsets. So for this one we have a control condition and we have a walk condition, and this was imagining walking. And then at the bottom we have a set of contrasts that we want to return. So for these we give them a name and then we define the contrasts. The outputs of the function will go into the GLM folder. You can see we have these in both the data space and the brain space, so this is the new free surface space in this example. And then we also output the beta maps, the contrast maps, T-maps, and some files related to the design matrix. You can see the PGN of the design matrix on the bottom right here. This is useful for just checking that everything is working as expected. Sometimes you can catch some design errors. The next function is the task fMRI multi-run function. So for this, we need to create a folder called MVPA, and the function will expect in this folder a bold image for each run of your experiment and a corresponding design file. So here you can see we've named them run 1, 2, 3, 4, and also for design run 1, 2, 3, 4. And the function will match these together. Again, we also need our reference anatomy, and instead of having the contrasts in each of the design files, here we give it a singular contrast text file. And you can see an example of what would be in that contrast text file at the bottom, where we have a name for the contrast and the definition for the contrast. And when we run this function, again, we get the same outputs as single run GLM, and we can overlay these statistical maps on our high resolution anatomy or on our lower resolution anatomy in bold space, or even on the bold image itself. So we also have a function for doing retinotopy if you collect retinotopic data at the scanner. We set this function up by creating a retinotopy folder, and it will place its outputs into a retinotopy out folder. We do it this way because, generally speaking, we will do retinotopy for the left hemisphere, and then we'll do it again for the right hemisphere, so we need multiple of these outputs. Into the retinotopy folder, we place the retinotopy data, the resliced co-registered data, and a design text file or JSON file. So here the pop-up will ask us for the primary condition and the number of lags for the lag-based correlation analysis. So this will be specific to the version of retinotopy that you run, but what we put in here will define whether or not we're looking at the left or right hemisphere. And supporting documentation for this will be on our ICE wiki. But the outputs will go into the retinotopy out folder. The primary one is the lag core phase in degree image. There are a number of different other images that are currently output with this function. Most of these are just for testing purposes. But when we overlay this lag core phase degree image on an inflated cortical surface, you can see that the definitions between V1 and V2 and V3 become very easy to spot, and we can map these out on our inflated cortical surface and bring those regions of interest back into volume space. For multivariate pattern analysis, we have a function that runs on the MVPA folder. The setup for this is very similar to the multi-run general linear model. The difference here is that we have this Weibo's text file where we define how we want the conditions within our design text files to map into Weibo's for multivariate pattern analysis. So you can see for this one where we have a number of different conditions, bedroom to office, bedroom to kitchen, games room to kitchen, etc. We map these into two different labels for pairwise comparison, or pairwise multivariate pattern analysis. And the output of this function is the CV results JSON, where we get out single trial decoding accuracy, average trial decoding accuracy, per run single and average trial decoding accuracies, and also single trial confusion matrices. So next we'll look at the functions we have for diffusion MRI. The schematic is an overview of how we typically go about this workflow. So we'd start by correcting distortions in the field using top-up, and then we do eddy and motion correction. Then we move on to tensor fitting, and if we wanted to do tractography, we'd do bedpost X for FSL's probabilistic tractography. So the way we've implemented this is that top-up, eddy, current correction, and motion correction, and tensor fitting are all part of a single function. This is the run DTI fit function. So this will target the Nifti resource of a scan and put its outputs into a DTI fit folder. And what we need to do for the Nifti folder to set this up is the Nifti folder for the diffusion scan will already have the Nifti file, the JSON, the bval, and the bvec files. But into this, we also want to place a brain mask for the diffusion image, and we name this no-diff brain mask. And then we also want to put in a top-up run we've collected for the diffusion. So the diffusion, if it's collected in an anterior-posterior phase encoding direction, we will have a top-up image in the posterior-anterior phase encoding direction. And we'll place this into the Nifti folder. And then once we run the DTI fit function, it will do top-up correction, eddy current motion correction, and tensor fitting. The input images will be stored in the DWI folder of the DTI fit resource, but the primary output will be in the DTI fit folder. And this will include things like mean diffusivity, the fractional anisotropy files, the L1 and the L123 and V123 files. We also output diffusivity along the primary sort of cardinal axes, the X, Y, and Z axes. And within the DTI fit resource folder, we also put some additional information files related to eddy current correction and quality assurance on top up. So once we have the DTI fit folder, we can then run the bedpost-X function as a prerequisite to doing tractography. This will target the DTI fit folder and create its own bedpost-X resource folder. And then within this folder, once it's complete, we'll have the bedpost-X directory and all the files that are prerequisites to doing tractography. So when it comes to doing tractography, the diffusion tractography function will target the bedpost-X resource folder and place its outputs into a tractography resource folder. There are a few things we need in this bedpost-X folder to do tractography. Because tractography is quite, there's quite a lot of different analyses you can do. What we want to do is we want to give it a specific set of files to follow. So we want to upload into this bedpost-X folder an anatomy.7z or an anatomy.zip folder containing our anatomy, our brain anatomy, either free surfer or native brain, and the free surfer ribbon output in the same space. So this will contain the white matter segmentation, and it will also contain the grey matter segmentation. And we use these for registering regions of interest into the diffusion space, or vice versa. We also want to include a seed masks compressed file. So this can contain a single seed segmentation, and this will be used for tractography from the seed region to the rest of the brain. If we're looking at tractography between multiple regions, we can put multiple seeds into this seed masks zip file, and the output will be related to tractography between these regions. Or if we want to do tractography between a seed and a target, we can include a target mask zip file where we place the target area that we're interested in. So for any of these combinations, when we run tractography, the output will be in the _work folder, and here we get the tractography paths in the free surfer space if we're using free surfer, or native brain space, and we also get the paths in the diffusion space. An important file that we also need to include for bedpost-X when we're running tractography is the tractography config file. So here are some examples of how that config file is set up for single voxel to rest of the brain, or single seed to rest of the brain, multiple seeds, tractography between multiple regions of interest, or from a seed region to a target region. And you can see some of the output here with the paths, the probabilistic tractography in the high resolution anatomical space. So next we'll look at the workflow and functions for doing resting state fMRI. This schematic is showing a sort of workflow we might want to implement for doing resting state. We'd start again by doing top up to correct for field distortions within our images. Again, we'd do some quality assurance. We would do resting state specific pre-processing, register our bold images to our anatomy, and then we're ready to do some analysis. Here it's connectivity analysis. So top up, quality assurance, and co-registration are the same as before. The unique step here is the resting state specific pre-processing. So the RSFMRI run pre-processing function will target the Nifti folder of a scan, and it will place its outputs in a pre-processed resource folder. The Nifti folder, we want to set that up with the bold images for our resting state run. We also want to include a segmentation for the left and right ventricles, and also the ribbon white matter. So these are used for creating confound regressors when it comes to denoising our resting state data. And for this, we also want to place the anatomy image from which these segmentations were derived. So we assume that for resting state, we're always going to be doing slice time scan correction, motion correction, and temporal filtering, or bandpass filtering. But here, the step that we make available to modify is the smoothing step, so we can either include smoothing or not, and then we can adjust the smoothing kernel. So again, we get a number of different outputs in the preprocessed folder. The main one we want to proceed with is this func_denoised_bandpassed image. And this would be the image we would take on to doing something like connectivity analysis with the RS fMRI connectivity function. So this function again targets a connectivity folder that we have to create within the scan. And within this folder, we will place our resliced data. So once we register it to the anatomy, we place this resliced data into this folder. We would also place a seed region for where we want to start our connectivity. So in this example, we've taken from the APARC 2009 plus ASEG Atlas an area of occipital cortex. And for the target, we have chosen the whole brain mask. So essentially we're doing connectivity to the rest of the brain. And within the raw connectivity JSON file, we have this JSON schematic defining these different parameters, and we've also turned on Fisher-Z transformation and an output prefix for our output. So once we run this function, we get the output within the same resource folder. Here we have an image showing the connectivity between the seed region and the rest of the brain, both the Pearson R correlation value and the Fisher-Z transform value. And we can overlay these maps on top of our anatomy. The final function to talk about is the function for doing quantitative susceptibility mapping. So this function is set up to do QSM for multi-echo GRE images. So typically for this, you would have two scans, and one scan would include magnitude images and the second scan would include phase images. The setup for this is to take one set of those images and place them in the Nifti folder of the other set. So here we've taken the phase images and placed them in with the magnitude images. And we can then run this function on the scan that will target this Nifti folder, and it will place its outputs into a QSM folder. So the outputs we're interested in is the chi map, that's the QSM image, but we also get the R2 star map and the T2 star map as part of this output set of images. This is an example of what the chi map will look like. There are a few additional functions that we have available. So we have a function for isovoxeling a scan. Here we create an isovoxel folder, place into it our image, and we can run this function that will target that resource folder, and we can specify the X, Y, and Z resolution we want to change to. We also have a function for modifying regions of interest. So if we have two regions of interest, we can either take the intersect of these or we can combine them into a single region of interest. The input fields are partial string matching, so as long as the target resource folder, the Y folder, contains two or more regions of interest, we can then modify these by partial string matching them within this function.